So I started working in the exosome field. I think what I would say probably right is an inception in like 2015. So this idea of extracellular vesicles, that idea has been around for a very long time. These are known molecules, secreted vesicles released from multiple different cell types, and this idea of them being carriers, traffickers of information, or even potential waste removal mechanisms that has been well studied in the cancer field. But in the context of neurodegeneration, it had not been fully explored. So us in the field were first trying to figure out ways could we first extract these extracellular vesicles from human plasma, because we obviously wanted to be some sort of, biomarker to assess what is happening in the CNS. Or I guess I should say a surrogate. That's the word I was looking for. And so probably a good chunk of the field was just optimizing the techniques, right? Because these extracellular vesicles, they're lipid-based molecules. They're very sticky. So they're known to kind of aggregate amongst themselves as well as traffic other molecules along with them. So we had to find ways in which we could actually optimize those techniques to where we could extract intact exosomes that had some sort of CNS specific origin. And once we had kind of optimized that, then the next thing was to characterize their cargo. So the vesicle, the extracellular vesicle is the shuttle system, but what we care about are the cargo that they contain. And so we have been pretty successful, myself and others in the field, at characterizing changes in AD-related proteins, including amyloid, tau, synaptic markers. People are looking at alpha-synuclein and TDP-43, and we're able to see that changes in those cargo levels, so in the amount of protein that is contained within these exosomes, does change in relation to disease state. And each marker, their profile changes. So some of the synaptic markers may be really low in a control patient, but then they may end up being very high in an AD patient, and then vice versa. So we've been taking our time to not only identify what markers are contained in these exosomes, but then what are their expression profiles and how do they change in relation to disease state. So I think the word is that there's a lot of promise in this field, but we're still trying to kind of narrow it down as to what's going to be the most suitable marker and what's going to give us the most consistent readout.